Well, hello everybody. Welcome to your very first lecture of Bio MO1. I'm Professor Thomas, and I'll be making a lot of recordings throughout the semester. And I have a serious caffeine addiction, but I acknowledge it, which is the first step to recovery. All right, so anyways, what should you do during these lectures? Well, I think that one of the biggest flaws of online courses, nothing wrong with the online course, but it's been a factor of online courses since the beginning of the online course, is that students think that it's going to be easier. I have news for you. It's not going to be easier. When I was in college, there was something called a telecourse. The idea behind it was that you buy tape recordings either on CD or, dare I say, VCR, which I know many of you do not understand the concept of the VCR. Suggest you search for it on Wikipedia. But the idea behind it is that you would watch videos, you would take notes, then you would come into campus for the midterm and final. Like I tell you, I took a telecourse class and I didn't watch one video and I got destroyed on the final or the midterm. Final wasn't too much better. You think I would have learned my lesson and watched the videos, but gosh, they were so boring. Like I'm talking like I watched one video and gosh, that was rough. But anyways, technically I didn't attend one lecture. Technically I didn't even deserve to pass that class because I didn't even put in the minimum amount of time needed to succeed in it. So that was pretty hard. On the other hand, as I progressed more through my college career, we had fully online classes where we could actually take tests online and things like that. That was a bit better. I still didn't really read the materials. And the end result is that I actually did worse in online classes than I did in in-person classes. So I finally kind of caught on that online classes are not going to be, were not the easy ride that I thought that they would be. So actually, if you're coming into this class thinking that it's going to be easier, then it's going to be a very difficult semester for you. And I find in general, so far teaching online, it's been a it's been a very interesting curve. There's the students that get A's and B's, and then there's actually not a lot of students getting C's, but there are a lot getting D's and F's. And you're probably saying, "Oh my gosh, professor, are you that much of a hard-nosed teacher?" No, they just aren't doing their work. They aren't doing their work. They aren't studying for tests. Like I'm talking about. During the test, you'll see that there are free response questions. Now, these free response questions are not torturous. In fact, if you ask nicely, I'll probably give you the topics beforehand. But I have students that they wouldn't even attempt to answer them. One of the things that happened last semester is one of the questions I had on the final was describe 3 ways that you can be more environmentally sustainable. And I had some students plagiarize it. Like they actually, because at the one campus I teach at, they don't let you do um, um, uh, any test proctoring software. But yeah, they copy and pasted it. Like 3 ways you could be lead a more sustainable lifestyle, and they copy and pasted it. So they technically plagiarized an answer that I didn't think anyone would have to plagiarize. Or on the other hand, is you have some students like this is what happened with one of the classes I taught last semester. Is I for the lab quizzes, you don't have lab quizzes, which you'll thank me for because you have Labster. But these lab quizzes, like I gave 2 attempts, meaning your or your highest score stuck. I can't tell you the students that only took it once. They might take it once and get a 6 out of 10, and they won't take it again. And the question is well, why? Because online classes, it's hard to get motivated for them. You have to keep a strict schedule. What that means is that you should be setting on your calendar or whatever. You should I'll be setting, okay, well, I'm gonna study, or I'm gonna have from 7 to 8 o'clock at night. That's going to be my time to watch bio lectures, and I'm gonna make it one day a week from 9 to 10:30. That's when I'm going to do my lab. I'm telling you guys, if you don't have a schedule, it's going to be hard. So this class, if you put in the time, watch the lectures, complete the outlines as you're listening to the lecture, put in your due amount of time studying. I give you use study guides. Then you'll do fine in the course. But if you don't, it's going to be a struggle. So make sure that you are prepared to put in the necessary amount of time. If you do, you'll succeed. If not, then it's, as I said, it's going to be hard. And you know what, for some people, maybe this may not even be the right time to take a college class. Right now is a very stressful time for a lot of people. So anyways, keeping that in mind, that is my speech. But know that even though I'm not the fastest email responder in the world, I will get back to you within a day and a half. So if you need anything, you can always reach out. But other than that, we got to learn some biology. So we're going to start here with, oh, and I have so I'm actually really proud of this. I have a wireless drawing tablet, so I can like draw and write on this tablet here, and it'll show up on the whiteboard. I do have to warn you though, my handwriting and drawing is not exactly going to win any awards for written excellence, but I try. All right, cool. So let's go to the whiteboard. All right, so biology. What is biology? Well, biology is not one class. What am I saying? It's not one topic. It's an umbrella term for a bunch of subtopics that are generally pretty different from one another. All right, drawing tablet. Now's the time to shine. It's actually an umbrella topic for a bunch of subtopics. So biology has a pretty simple definition. It's just the study of living things, the study of life. Chemistry, on the other hand, is the study of chemical processes. And here are the major tenets of biology that we'll be talking about. So the first is going to be cellular and molecular biology. So this is things that are at a microscopic level. And this includes things, for example, like things like energy systems. So for example, photosynthesis, cellular respiration. So that's probably the biggest one that we talk about in this class. We also have trying to see how to phrase this. I'll just put down cellular organelles like mitochondria, rough ER, you know those terms. We also have communication. Cell communication is a big part of biology. I don't know how much your textbook, it's actually my first time using this textbook. I don't know how much your textbook's going to dive into that, but it could be quite a bit, so might as well write it down. And genetics. So cellular molecular biology is one of the biggest tenets of biology. Okay, well, there's more. We also have ecology. Some examples of that are going to be animal behaviors, population dynamics, which is interesting because it's what determines the population of animals in a certain area. We also have food web / chains. And of course, there's more. I don't want to bore you guys too much. You're already probably saying, wow, this class is really getting my blood pumping. All right, we also have evolution. And this is the study of species relatedness. Evidence of evolution. Types of evolution. I know. Evolutionary science is a bit controversial. I know that there's some thought that it's not really a science, it's more of a pseudoscience because you can't exactly reproduce this in a lab. Well, you kind of can. Yeah, it's complicated. All right, going to clear this out here. You also have some more. So ecology, evolution, and cellular molecular biology is probably the things we're going to spend the most time in this class. But we also have some other terms. We have biotechnology. And this is the study of combining genetics with computers. So this is things like gene sequencing, genetic modification, which is probably the biggest one. Genetic modification, DNA fingerprinting, which is you can actually take DNA at a crime scene and sequence it and match it with a suspect. DNA fingerprinting, paternity. So you should probably just know some examples for the quiz / test coming up in a couple weeks. All right, we also have environmental biology. And this is going to be the study of man's impact on the environment. Yeah, I teach environmental science at the high school I teach at. And the first thing I tell the students is, throughout this class, be prepared to have humanity just ragged on throughout the entire semester, because humans are the only species that have ever walked the earth, and we're talking over hundreds of millions of years of life being on earth, or maybe even longer, that's had a net negative effect on the earth, meaning that we haven't contributed to it at all. And of course, one of my favorite topics, which, no offense guys, but I'm kind of bummed I'm not teaching it this semester, is physiology and anatomy. This is actually the study of the human body. I'll just put it at that. So let's put down structure and function. I know it sounds easy, but if it was that easy, there wouldn't be an entire course on it with stressed out students that send me very emotionally charged emails. We'll just put it at that. All right, so that is biology. The major tenets of it, we'll be talking about these in varying quantities throughout the semester. So now we're going to talk about, because biology is a science, we do need to talk about the scientific method. scientific method. And you're probably saying, okay, well, what does the scientific method mean? Okay, that's a pretty interesting C. But yeah, scientific method. What is the scientific method? It is just a series of steps to determine the truth. And that's because humanity's greatest enemy in determining the truth is called bias. What is bias? Well, bias is when your ability to find the truth is clouded by past experiences and personal preferences, relationships, so on and so forth. For example, if I were to ask you, which fast food has the tastiest burger? Oh, well, obviously it's In-N-Out. Now, why is that? How do you know? Well, chances are is you didn't do a blind taste test where you laid out the hamburgers on the table, all with the same condiments on them, all at the same temperature. You probably didn't rinse with water between each bite. No, that would be good science. Instead, you're probably thinking, well, I think that In-N-Out tastes good. All right, great, but why? What makes it taste good? So it could be because you have really fond memories of going to In-N-Out when you were a kid. Maybe you went there every day after sports practice with your friends, and you remember the laughter and things like that. So it could not even be that the hamburger was the best, which they do make a pretty good burger. Don't get me wrong, but it could be your personal feelings toward In-N-Out actually cloud your answer. The scientific method is designed to eliminate bias, but it is still present. Bias will always be your greatest enemy toward finding the truth. So the scientific method is a series of steps that cannot really be skipped or compromised. Well, okay, they can kind of be skipped, but um, well, no. In this class, we're going to assume they can't be skipped. So the purpose of the scientific method is to eliminate bias. And bias is simply external factors that cloud the ability to find the truth. Of course, that's the goal, but it doesn't always happen because even the best, most experienced, most brilliant scientists, they have things that make them biased. It could be pressure from the funding company. Maybe, for example, McDonald's is doing a study saying, hey, find out who has the best burger. But we'd be very disappointed if ours wasn't. Okay, they just introduced bias. Or maybe it could be that you have a financial interest in having the experiment prove your hypothesis. So it is a number of factors. All right, so now that you have that down, let's go ahead and talk about the steps. So the steps of scientific method. By the way, I really don't like PowerPoint. On occasion, I'll post an old PowerPoint that kind of has some of the same material. But for the most part, I really don't use PowerPoint. If you're a PowerPoint fan or you have invested in the company, then I'm sorry. All right, so the first step, and this is how science always starts, is something called an observation. Okay, that's too ugly even for my standards. Observation. Now, an observation is something that you notice. Something you notice in the environment. It just is something you notice. So it could be something like, I noticed that In-N-Out Burger has the longest lines. Or it could be something like, I noticed that the best athletes wear Nikes. It could be something like, I noticed that more people buy iPhones than Androids. I don't know, but it just is something that you notice. And an observation always leads to what's called a question. And a question is the why behind it. Figuring out gosh, no, I don't like that definition. We'll just say that a question is going to be wondering why. Why does In-N-Out have the longest lines? Why do the best athletes wear Nikes? So on and so forth. Now the next is going to be a hypothesis. Hypothesis. And this is a statement based on prior knowledge, meaning that you just can't make a hypothesis that has no basis behind it. You have to have some knowledge on it, based on prior knowledge that predicts a conclusion. So it's very important that we clarify the hypothesis. The hypothesis is kind of similar to a thesis statement in a paper, except you aren't trying to prove it right, you're trying to determine whether or not it's true. As a scientist trying to find the truth, you should be just as happy if your hypothesis is correct than if it is incorrect. The hypothesis is just a working statement. It does not necessarily have to be true. It just is a working statement. You're going into the experiment thinking that this is going to happen. And generally during a hypothesis, you predict the effect of one variable on the other. So for example, the reason why or a In-N-Out Burger is so popular because it is the tastiest, or most athletes wear Nikes because it helps them run faster. So those are examples of hypotheses. Next, we have the experiments, and we're going to be talking about the experimental variables in just a moment. Then we have the analysis. And the analysis is interpreting the results in a non-biased manner. In the past, I used to tell students, think of it like a news report that's just trying to tell you the truth. However, now the media has shifted more to a model where they attempt to incite your emotions instead of giving you the facts. So I can't really say that anymore. But what the purpose of the analysis is, and if you ever read an analysis in a research paper, it's going to be very matter of fact. Is they're just trying to explain to you what the experiment or what the experimental data show. And next you have a conclusion, and a conclusion is the interpretation of the results and how it relates to the hypothesis. And after a while, an experiment could lead to a theory. And a theory is when multiple experiments show the same results. That means if 20 different research groups do the same experiment and they get consistent results, then you can say that the hypothesis becomes a theory. All right. Okay, so the experimental variables is going to be a little bit down here. So what can we apply the scientific method to? So now we have scientific method, applications. So what can we apply the scientific method to? Mainly, it is things that are quantifiable. So these are mainly things like mass. You can't dispute mass / weight. Be things like height or size. Be things like reaction rates. Can be things like numbers of something. So for example, in the vaccine studies, what they did is that they detected the quantity of antibodies produced after the injection of the vaccine, and that's how they know that it was effective. You can also use it for what we call qualifiable, and these are things like thoughts. And qualifiable data is not nearly as accurate because people can really be biased. But thoughts, feelings can also be opinions. It can be it can be statements. I don't know, even though statements is kind of similar to opinions. So whenever you get a survey in your email saying like rate your satisfaction, that is they're asking you for qualifiable data, but it isn't as accurate. Ideally, the most accurate data will be from quantifiable. Now, what can't the scientific method be applied to? And it's things that unapplicable. Mainly, it's going to be the paranormal. You can't measure the paranormal. And the other is going to be beliefs. You can't apply the scientific method to a belief. Now, does this mean that there's no paranormal activity? Does this mean that there's no beliefs? The answer is not at all. There could be paranormal activity. Your beliefs are perfectly valid, but the scientific method just can't be applied to them. How are you going to prove the paranormal? You can't. Not with the scientific method. Now there are things that might make you believe in paranormal activity. There are and of course belief systems are very personal and near and dear to your hearts. But you just can't apply the scientific method to them. That's all. Kind of like saying you can't use peanut butter as a band-aid. But peanut butter has lots of other applications, like it does really well on sandwiches, even celery. Now that also speaks to our two types of data collection, and that's exactly what I have here. Is quantifiable is anything that can be measured in numerical quantities. And as I said, it is the most accurate type of data. It's anything that can be, that's not a Q. It's anything that can be directly measured. And it generally is going to be indisputable, meaning that you can't argue with it. You can't argue with mass and weight. You can't argue with height. You can't measure with reaction rate. You can't argue with numbers of anything made, like antibodies or a certain product. These are things that you can't argue with. Temperature. Qualifiable, though, has to be indirectly measured. With a scale, meaning that you can do rate from 1 through 10. That's probably the easiest way to do it. Or you can interview people and record how many times they say a certain word or statement. But as I said, is people can be biased. For example, one thing that they noticed during the first stages of determining the symptoms of SARS-2, COVID-19, is they interviewed people in Wuhan asking what their symptoms were, and there's actually a very low percentage of people having a symptom of diarrhea or digestive issues. And the reason why is that they're actually ashamed to admit that they were having diarrhea. So it was a symptom that was underreported because people were embarrassed by it. So what that does is shows you how qualifiable data is not always accurate, because it is the most prone to bias or personal opinions. And yeah, qualifiable data at times needs to be taken with a grain of salt. Maybe in opinion survey, maybe if somebody's having a bad day, they'll rate things harsher than if they're having a good day. All right, so I encourage you all to take a couple-minute break, and then we're going to finish this lecture. So I'm going to go ahead and clear this out, and I'm going to encourage you all to take a, I would take a 5-minute break, just because it's hard to sit down for long periods of time. So I'm going to go ahead and pause this recording, and I would like you to just get up, walk around, do some jumping jacks, push-ups, whatever. See you in about 5 minutes. All right, well, I hope you enjoyed your break, even though it seems like there was no break because I just paused the recording, then came back. So it's like, all right, well, came back in 5 minutes, and Professor was back in 5 seconds. I probably should have told you to pause the recording, but oh well. By the way, you can actually watch these recordings at 1.2 speed if you want, because you really won't miss much. There's not really too much of a difference between 1x speed and 1.2, and it might help you get through it a little bit faster if that's your preference. Well, actually, yeah, 1.2 is good. It's probably a good idea to pause the recording when you need to catch up on notes. You'll figure out which way works best for you. All right, so now we have experimental variables, which is one of the most important things to grasp, but also one of the most difficult, because this is not something you're used to doing. But anyways, experimental variables can be broken up into two types of categories. And one is going to be what we call independent. And the other is going to be what we call dependent. Now, an independent variable is anything that the researcher determines. The researcher has complete power over the independent variables. And there are two independent variables. Actually, let me slightly adjust this. Just so I have enough room to write. Independent. So there's two types of independent variables. One is called an independent controlled variable. Do not confuse this with control group. This is a controlled variable. And the other is going to be an experimental variable. A controlled variable is anything that stays the same, between experiments. And I'll give you some examples of that. And an experimental variable is altered during experiments. Now, why is that? The experimental variable is the one you are focusing on. You are trying to see the effect of the experimental variable on the outcome. Let's say you're trying to determine which dose of vaccine result in the greatest number of antibodies made. What you'll do is you'll have different groups that receive different amounts of the vaccine. The quantity of vaccine differs between the groups, therefore that would be the experimental variable. And we'll talk about the controlled variables. Controlled variables are things that stay the same. Ideally, you want all of your experimental groups to stay the same except for the experimental variable, as you want everything else to stay the same. For example, in this example I used for the controlled variable, you would want the same demographic patients, so varying age groups. You would want the vaccine given by the same doctors. You would want the vaccines to go into the same place in the body. You would want the vaccine to be I mean, this is kind of going a bit too far, but you would want to make sure that the vaccine is given, that all the vaccines are kept in the same temperature. And you're doing this because you only want to see the effect of the experimental variable, nothing else. The dependent variable is going to be the outcome. It is going to be the outcome. And the dependent variable is basically the results. So it's the outcome / results. So if your hypothesis is going to be that the, I don't know, 50 milliliters of vaccine will give the highest quantity of antibodies, then the quantity of vaccine would be the independent variable. The quantity of antibodies would be the dependent variable, because that's what you're trying to find out. You're trying to find out the quantity of antibodies. We'll go over another example after. Now, there are two types of experimental groups. Two types of experimental groups. And the first is going to be what we call the control group. And a control group is not exposed to the experimental variable. And its purpose is to be a comparison group. How do we know that the vaccine is effective? How do we know that? We just can't give people the vaccine and measure the amount of antibodies, because we don't even know if the vaccine works. For our control group, in this example, it would be people that do not have the vaccine. And what you would do is you would have these people. They would go out and live their lives as normal, but they would not have the vaccine. And the ones that unfortunately get sick, you would measure the quantity of antibodies that they make, and then you would compare it to the quantity of antibodies triggered by the vaccine. Now, let's say that the vaccine results in a quantity of 50% more antibodies than in the control group. Then you would know that the vaccine is effective at making more antibodies. You could also do quantity of or number of people that become symptomatic. So the control group, you can say, okay, out of 1,000 people, 250 develop COVID-19 symptoms. Meanwhile, in the vaccine group, only 5 people develop symptoms. So therefore, you could also use the rates of a positive test in order to determine whether or not the vaccine works. So your control group is your comparison. And the other side of it is going to be the experimental group. Experimental group. And this is the group that is exposed to the vaccine. Exposed to experimental variable, hence the name. Okay, so if you want an example, we can go into it like this. So I'm going to say that my observation is going to be plants indoors grow less than plants outdoors. Of course, this is a topic I'm sure many of you spend hours wondering. My question could be, why do plants indoors grow smaller or grow less? So that will turn into my hypothesis. Indoor lighting is not as effective as outdoor lighting for plant growth. Okay, if I'm going to create some data here, I'm going to go ahead and erase this. My controlled variables I would want the same type of plant. I would want the same experiment duration. That's not how you spell experiment, but you get the idea. Same experiment duration. Same room. Same researcher. So you want everything to be the same except for one thing. The other side, we have the experimental variable. And this would be type of indoor lights. And we could have incandescent, which is your normal light bulb. We could have, I'm going to spell this wrong, fluorescent. We could have LED lighting. That's not, that says LEO. We could have LED. There's also, gosh, I was thinking on types of light. LED, we'll just do, I don't know, iPhone lighting, whatever. flash. Oh gosh. Handwriting is not on point today. And our experimental group. So the experimental group is exposed to indoor lighting and our control group is exposed to outdoor lighting, outdoor sunlight. Because we're trying to see if indoor lighting really is less effective at making plants grow than sunlight. Well, so what we would do is we would see how much the plant grows when exposed to sunlight versus when it's exposed to indoor lighting. And that would be our results. Now, for the analysis, you would say, for example, sunlight resulted in 12 centimeters of growth, incandescent resulted in 8 centimeters, fluorescent 6 centimeters, LED 2 centimeters, iPhone flashlight 7 centimeters, and you would just give the raw data. For your conclusion, you would say, okay, well, outdoor lighting made the plants grow 18 centimeters. Therefore, it is the best type of light if you want plants to grow optimally, which shows that my hypothesis is correct. And then if you do this a bunch of times, then it can become theory. All right, so now we're going to move on to levels of organization. Going from largest to smallest. And the first one is going to be biosphere. And what this is the Earth and everything in it. Next is going to be a biome. And a biome is an area with consistent climate and plant animal life. So for example, a tropical rainforest. It has a very consistent climate, hot, humid, rainy, windy, and you can expect to find similar plants and animals in the rainforest. Next is an ecosystem. And an ecosystem is life forms interacting with their environments. For example, a lion chasing an antelope, or a lizard soaking up heat on a heat rock, or a groundhog burrowing in the dirt. So it's life forms interacting with their environment. Next is a community. And this is life forms interacting with each other. Interacting with each other. So a bunch of rams fighting over mates, mice sharing food with one another, a zebra being chased down by a tiger. So it's any times that life forms interact with each other. Next, we have a population. So I'm going to go ahead and erase this. Next, we have a population. And a population is members of the same species in the same area. So you are a population of students at Moore Park College. So populations are great for making comparisons. What is the size of mice in California versus the size of mice in Nevada? Organism is a singular life form. And now we're going to break it down into components of a life form. We have an organ system. We talk about these a lot in anatomy physiology. Organ system is organs that function correctly, that function together to complete a task. For example, the digestive system is a group of organs that work together to break down food and absorb nutrients. The renal system works together to filter blood and maintain proper blood volume. An organ is a structure. Organ is a structure of several tissues that performs a function A tissue is a collection. I don't like that. It is comprised of same type of cell, like muscle tissue, or adipose tissue. A cell is the smallest unit of life. Smallest unit of life. So yes, a bacteria, for example, is a singular cell. A protist is a singular cell. A virus is not a cell. In fact, viruses technically aren't alive if we go by our criteria on the next page or section. Then we have molecules, structures comprised of different types of atoms, different types of elements. And an atom is the smallest structure that comprises life. Talk a bit about atoms next lecture. All right. So these are the different levels of organization from largest to smallest. And now we are going to talk about the characteristics of life. So now we have the characteristics of life. The first one is going to be cell. All life forms are comprised of cells. The cell is the smallest identifiable unit of life. We'll talk more about cells in the subsequent chapters, but it is the smallest identifiable unit of life. Next is reproduction. And reproduction is the ability to copy DNA or RNA. Again, we'll talk about DNA and RNA later on. Copy DNA and RNA and pass down the offspring. And yes, even bacteria can reproduce through a process called binary fission. Then a life form must have the ability to sense, which is to receive and interpret environmental stimuli. Yes, bacteria can do this too through a process called quorum sensing. Then we have respond to the environment. Respond to environment is actions caused by environmental stimuli. And yes, bacteria can do this one too. An example of you responding to the environment would be if you're walking down the street, just minding your own business, and all of a sudden you see a grizzly bear just like walking by you. Well, what your brain's going to do is it's going to cross-reference the picture of the bear with all the documentaries you've seen on TV and also scary movies, going to determine that the grizzly bear is a threat, and you're probably going to book it, scream and run, or if you're very brave, you might attempt to take on the grizzly bear mano a mano. That's an example of responding to the environment. Homeostasis is a complex term, but we'll go ahead and agree on this definition of homeostasis. It is the ability to maintain consistent levels and states. So, for example, an example of homeostasis is that we maintain an internal body temperature of around 37 Celsius. If our temperature gets too high, we start to sweat. If temperature gets too low, we start to shiver. So homeostasis is the ability to maintain consistent levels and states of being. We'll go over more examples later. The homeostasis, ability to maintain consistent levels and states of being. And last is evolution. And that's the ability to change over time. All life forms evolve, well, according to the theory of evolution. Change over time due to, it's kind of hard to, due to changing environmental conditions. This is really only a half truth, but it will do for now. Changing environmental conditions. Like you'll notice that polar bears have a white coat, where brown bears have a brown coat. Why is that? Why did one common bear species diverge into the polar bear and the brown bear? And it's because something happened. They got separated, and the bears changed their genetic code over time. Well, not consciously, but we'll get into that later. But they changed their genetic code over time until eventually one bear had a light coat, one bear had a brown coat, which helps them survive in their environments better because a polar bear can blend in with snow and a brown bear can blend in with trees. Okay, so this is the end of chapter 1. Now, what is your task now? Well, now you can go back, watch this lecture again if you choose. Make sure that your outline is filled in and that you've taken adequate notes or exceptional notes. You also want to go and do the chapter 1 discussion board questions because those are graded. And you want to look at the study guide, make sure that you understand the concepts you'll be assessed on, and then it's time to watch the chapter 3 lecture. Chapter 2 is kind of a continuation of chapter 1, so we're going to go ahead and skip that, and we're going to go on to chapter 3. All right. Any other questions? Reach out. If not, see you during the next video.